Welcome to Careers in Discovery, where you'll meet scientists who've forged outstanding careers in biotech and hear about what they've learned along the way. This podcast is brought to you by Singular, building brilliant biotechs. Matthew Duchars is the CEO of CHAIN Biotech, a fascinating company developing oral vaccines and immunotherapies that target the microbiome. Matthew joined us to discuss the importance of culture and environment in delivering great science, how a career in vaccine development was set into motion by an encounter with a dead mole, and being at the heart of the UK vaccines ecosystem during the COVID pandemic. This week on Careers in Discovery, I am delighted to be joined by Matthew Duchars of CHAIN Biotechnology. Matthew, welcome to the show.
Thank you, Tom. Thanks for inviting me to come on.
Really looking forward to talking to you today, Matthew. To start off with, I wanted to talk a bit about CHAIN and the work that you're doing there. I mean, the microbiome, of course, has risen in prominence over the scientifically speaking. It's always been important, obviously, but scientifically speaking, over the last, I guess, 5 years particularly, right? There's been a number of discoveries that have accelerated that. But tell us a little bit about the approach you're taking at CHAIN and the work you're doing around the microbiome and bacteria to treat disease.
Yeah, thanks. So I joined CHAIN about a year ago, and one of the things that sort of attracted me to CHAIN was that it had a very unique approach to its technology, and it was this combining the microbiome that we talked about, but using them in an engineered way to be able to deliver sort of oral therapeutic vaccines. And there are several sort of aspects to that approach that were quite attractive to me. I've spent probably 30 plus years working in vaccines now, in developing vaccines. And there are several things that we have still sort of eluded a lot of vaccine developers and are sort of seen as the holy grail. So being able to deliver vaccines orally, not have to use the needle in arm, is one of the things. Stability has always been a big issue as well, and we've seen that recently with some of the great progress that's been made with things like the RNA vaccines, but they had to be kept ultra cold at -80.
Yes.
And one of the approaches or the approach that we've taken at CHAIN is we've harnessed a natural safe probiotic. It's got a long track record of use. And have found a way to be able to engineer antigens into that strain. So we use a single strain. It's not like a whole array of different strains that are quite often found with microbiome treatments. But this is very specifically focused on one particular strain that has this long track record of safe use, and we've been able to engineer into that different types of antigens, and found ways to be able to express antigens in multiple ways. So either intracellularly, extracellularly secreted, or on the surface, cell surface. And that presented some real opportunities, I think, in the sort of vaccine world for us. Probably the most at the sort of heart of it, the most compelling piece of this is that you almost get sort of 3 shots at the target for one, because it has almost a sort of triple mode of action in terms of how it works. So not only does it enhance the microbiome, it produces natural metabolites that improve the sort of gut homeostasis, but it also has these antigens that we can express and produce, and that helps with a more localized response in where they're expressed in the lower gut. So you get sort of a mucosal immunity response, and then also a systemic response as well. So you've got sort of 3 different approaches that have been taken through the one technology. So that's sort of at the heart of how it works. But in addition to that, there are several other things, as I mentioned, that are quite compelling about the technology. The fact that it is orally administered, so you just pop a tablet swallow a solution, which is much more preferable to generally to having a needle stuck in your
absolutely.
arm. And it's very very stable as well, because it's based on Clostridium spores. So the spores are a bit like seeds. They sort of are dormant, very resistant, and can be essentially sort of stored at room temperature on the shelf. They've got longevity. You can come back to them 5, 10, 15 years time, and they'll still be active. So you don't have all the issues about having to be able to store them at ultra low temperatures, and how long will it last for. These things are like sort of stable brick dust. And then when they go into, when you actually sort of take the therapeutics, take the spores, they travel through your gut, and then where they would naturally start to grow is in the lower intestine, in the colon, which is a very anaerobic area where Clostridia naturally occur, and this is, as I said, a naturally occurring form of it. And then they start to grow and express the antigens of interest. So so that presents quite a different and unique way to be able to administer vaccines. And so that was very CHAIN's still very early stage. We're not in the clinic or anything. We're still preclinical. But I think there's some very compelling reasons why this technology could be really significant, with the big if it all works as advertised, which is what
of course
is all about. So it will come down to the science at the end of the day. So we'll see what we get to.
Yeah, absolutely. And presumably then, if you can make the mechanism work, there are then lots of other antigens that can be engineered into this. I know we don't really use the word platform anymore, but this approach, and there's lots of different uses that
no, I think that's exactly right. And so what we have done, and in fact, just recently we've had in the last year or so a slight change in our strategic direction in terms of what we've been working on. So we have done a lot of work previously on infectious disease vaccines, and that continues, and we're funded through Innovate to develop some vaccines via that route. But also we started to look at immune-oncology and being able to produce a sort of therapeutic vaccines. And one of the reasons why this particular approach is very attractive is there's been some quite recent papers in Nature, in fact, where Clostridium butyricum has been used in combination with things like checkpoint inhibitors, and it shows significantly improved enhancement when trying to treat different types of cancers. So we've got kind of a step up there already, because we're using Clostridium butyricum, and now we can engineer the antigens into it as well. So that really pointed us towards, let's take a closer look at this and see if we can actually develop an immune oncology based therapeutic, so for treatment of cancers. And we're focusing in the first instance, because you can't do everything, of course, but we're focusing on cervical oropharyngeal cancers, which are particularly caused by human papillomavirus, HPV. But this is about sort of treatment of those early stages of cancer. So it's not prophylactic. There's already good prophylactic vaccines out there for HPV. It's a different kettle of fish. This is much more about treatment of lesions and cancers as a result of HPV.
Yes, I see. Interesting. And I think that the whole topic is fascinating in that, as our ability to engineer biology in all the forms that takes increases, of course, it opens up areas that are completely unexplored in some of the advanced gene therapies and things like that. But I think one of the most exciting things is if you think about, obviously, the drug development process being so long and so costly and so high in failure and all this kind of stuff, the ability to leverage things that we know work and use them in different ways, I think that's really exciting.
Yeah. And I think one of the other things that's very compelling about this is, as I mentioned, it's already used as a natural probiotic and has been used for 30, 40 years. So there's a good, long, safe track record of use in humans. So that also provides a way of sort of de-risking the project, if you like, or the program, in that there's already ways to manufacture the material, and that it's got a good safety record already. So that gives you several advantages straight out the door.
Yes, absolutely. And we know biotech CEOs wear many hats, and those hats change depending on the phase of the company and the timing and all that kind of thing. What have you been mostly focusing on recently, Matthew?
Well, a CEO's work is never done because there's never enough money. So it's all about raising money.
No, of course. Yes.
But underpinning that, you can't raise money unless you've got good science and good data to back it up. So my primary focuses are on trying to ensure that we're heading in the right direction scientifically in terms of the data that we're generating, and that we can then use that to influence and persuade people to invest in us. And that then enables us to be able to do further work, of course. So that's always a big part of the role of a CEO. But then I think the other things that are important, and I don't want to lose sight of this as well, is the people and the culture that you create within a company. So that really determines how successful you are generally. And it's one thing I've learned over the course of my career is more and more I've got to realize the importance of the environment, the people, the culture that you've created and that you're working in, and that's what drives people. If it's a miserable place to work, nobody wants to work there. So they'll go in, they'll do their 8 hours or 7.5 hours or whatever it is, get stuff done. But if people are enthused about it and they love working with it, and the people who they're working with are good as well, then that really brings it to another level, and that enables you to get so much more done.
Yes. Yeah, and it's interesting you bring that up because, I mean, we think about it in a very similar way in that, of course, it's more complicated than this, but broadly speaking, there are 3 pillars to being successful as a biotech company, right? There's the science, the money, and the people. And the first 2 are so demanding that it can be easy to forget about the third one, right? But actually, the whole thing falls down without the 3 of them. Yeah.
Yeah, and that's absolutely. I think the thing that you learn as you go along, early on in your career, you're very focused on how I can get something to work or how I can sort of achieve things scientifically. And it's as you develop, you learn that actually it's you can't do all this stuff on your own. You need people around you, and in order to get the most out of people and to get that good sort of synergy, it's all about having people who get along, and people. Everyone doesn't love everyone all the time, but you still need to have an environment where people are comfortable, enjoy what they're doing, and enjoy where they're working, and have a sort of a focus and a meaning to what they're trying to do as well. So I think that's another important aspect in terms of what you're trying to achieve. I also found that out quite interestingly. Late on in my career, in one sense, I had a bit of a sort of seminal moment where I'd spent 25 plus years or whatever working in industry, always in sort of the commercial area of trying to generate a profit. And then I took my first job with a not-for-profit organization, the Sabin Vaccine Institute, and it was kind of a revelation, really, because I went in there and everyone was, just as worked just as hard, but they were driven in a very different way. They came into work because they were doing something that they knew was the right thing to do, and they knew that this was, had purpose to it, and that really sort of motivated people and really motivated me as well, actually.
Yes. Yeah, absolutely. And I think it's interesting, isn't it? If you've been part of a brilliant culture, nobody needs to explain it to you. if you haven't, it's very hard to explain.
It is. It
But the
sounds kind of
differences
corny
start.
because lots of people talk about, the importance of culture and we've got to get the right thing. And you quite often you see sort of companies, particularly larger companies, really sort of trying to force culture on people, and it doesn't work like that.
Right. Yes.
It just
No.
doesn't work like that, and it's really difficult
No.
to put your finger on what it is. And I mean, if it was that simple, everyone would be able to create fantastic culture and it would all be great all the time. But it doesn't work like that, and you have to have the right motivation, the right people who are driven in the same way, that there's a sort of similarity in terms of motivation towards a particular goal.
Yes.
All of these things sort of coalesce and bring people together to be able to achieve so much more than you can in a situation where you're not so excited about the people or where you're working.
No, absolutely. Absolutely. And I'm sure we'll come back to this as we talk about your career, Matthew. We like to take it right back to the beginning as well. And as you said, you've spent most of your career focused on vaccine development. There's also been, of course, a strong theme of microbiology running through your career too. But take us back to the start. Why science? Why vaccine development? Why drug development? Why this career for you, do you think?
Well, there's a question. I guess I've always had a fascination for science and how things work, particularly in biology, but in science in general. And as that sort of interest developed, there was a sort of natural progression towards trying to understand how things worked and then test out whether something could be improved and how that could work. And so that's always sort of underpinned my my sort of interest and direction in terms of what I've done with my career.
Yes.
And it's easy to you could say sort of why drug R&D or whatever, but it's not really been a part of any grand design, to be honest. It's it's been much more a case of doing things that you enjoy doing and that you find interesting, and taking opportunities when they come along. I mean, you actively look for them, but you also have to be able to sort of stand up to and decide that you want to take something, or take a bit of a brave decision. Sometimes it's not always comfortable. It's not sort of what you're used to doing. You have to go outside of your comfort zone quite often to try something new out, and it doesn't always work. But that's how you sort of find out what you are good at and what you do enjoy. So that sort of has been sort of the driver, I suppose, behind why I've followed a particular career. But if you look at sort of over the course of, I'm not going to say how old I am, but the things that have influenced you, and they're usually some sort of seminal moments along the way where you've had a fork in a road and you've had to make a decision. And I guess, if I sort of cast my mind back, that the first one of those actually all started with a dead mole. And this was when I was probably about 10, and at school, a couple of school friends, and we found a dead mole outside in the playground or the field or whatever, and took this back to our sort of science teacher, and I was just really curious as to sort of about the sort of biology of what had happened and things. And he said, well, why don't you dissect it? Why don't you have a look at it? And we sort of got there, and we had all these handkerchiefs and things around our faces, to sort of like we're sort of surgeons or something, and having a look at this, dissecting this poor mole. But that really sort of, I guess it was that encouragement from the science teacher to have a look and find, you want to know how something works. You want to know what's in it. So let's go and have a look. Let's go and try it out. Let's see what's in it. And that was sort of like my first, I guess, at least the first one that I can remember of me taking an interest, but why I particularly took an interest in science and the scientific direction. and then that sort of, that followed me through school, and I went, enjoyed doing my science subjects. I found that I had a sort of natural aptitude for it, which is also always, if you enjoy it, and you find that you tend to be better at things that you enjoy. So that's good.
Yeah, of course.
And then, you get to the end of your time at school, and it's time to look at whether you want to go to college, what you want to do there. And it's sort of, I don't know, I've never been a person who's like known exactly what I wanted to do. I didn't sort of, when I was 10 years old, and I said to them, I decided, that's it, I want to be a microbiologist, and I'm going to lead a biotech company. It's like, you don't know that stuff at the time. So what you do know is what you enjoy at the time and what you might want to do with it. So when I went to college, I actually did a very sort of broad foundation year, which was great because it meant I got to try out lots of different subjects, and found that some of the things that I thought I was going to really like, I really did not like. And some of the things that I had not thought about at all, microbiology being one of them, I actually found I really liked. And so that sort of led me down that path. And at the end of my degree, in your sort of final year, you do a research project, and I had really loved doing that. So I knew that I wanted to sort of continue doing that type of work, and that sort of led me naturally towards doing a PhD, and that served as a really good grounding for my future career in scientific research. And it's like any sort of part of any job or career, it's not a bed of roses the entire way. There are moments where you just think, what am I doing here? This is like, I'm here, it's still here at midnight pipetting things, and it's like, but you have to sort of take a step back and see sort of where it's going and what you like about it. And I really enjoyed my research. I enjoyed my time at Sheffield, doing my PhD there. But I knew by the time I got to the end of that an academic career was not the way for me. So as much as I enjoyed research and enjoyed making discoveries and finding out new things, my real sort of hankering, I think, was to try and find something where those findings could be applied to something. So that sort of pragmatic expression, a more practical endpoint. And academics are paid lousy anyway, so that also helps to get a bit more money. So off to industry I went.
And
that was actually my first job there. Was actually I joined what was at the time Britain's third biggest company, I think, Imperial Chemical Industries, or ICI as it was called in the day.
So you went to the large end of the scale straight away.
So that's which is kind of unusual. And my career has been populated from working in very large organizations, ICI, Zeneca, right down to a one-man band of sort of running my own business and just being the only employee. So being the grand title of CEO, but also the grand title of janitor as well, and change the toilet rolls and everything else. So quite a sort of spectrum there, and they all have their sort of pros and cons to them, I guess. My first role, as I said, was with ICI, and it was I joined at a very interesting time. So ICI had just come out of having developed a microbial protein food additive for animal feed called protein. They did it on an absolutely massive scale. It's still, I think, probably the world's biggest fermenter. There's a 1.5 million litre continuous fermenter, which is quite big. So they had done that, and it was a huge success, but commercially, unfortunately, didn't work out, and the economics of it, for various reasons, went against them. and so they had developed all this expertise in large scale microbiology, knew that protein wasn't going to go forward, so they were like, okay, so we've got all these scientists and all this stuff, well, what else can we do? And that was the point at which I joined the company. And so
I see. Okay,
that was a very important step in my career, actually, because the whole sort of purpose of after I joined, the next few years there were all about trying to find new concepts, new ideas, and how to translate them into something that is a practical, pragmatic solution that hopefully can make some money. And they've really thought sort of outside of the box, and were quite ahead of their time, actually. This was quite a few years ago. Worked on things like biodegradable plastics that were carbon neutral in the way they were produced. Nobody really worried about that at the time. It was like, you just go and get a plastic bag from Safeway. Why would you worry about developing a more expensive plastic that could biodegrade? Things like using enzyme technologies that were more environmentally friendly to bio-bleach paper. Using bioremediation to help reclaim contaminated land, production of vegetarian meat alternatives, which turned out to be corn in the long term. So now you can actually buy that in the shops. It's very readily available. But at the time, it was like, that was before it was still sort of in concept and under development. And people were like, well, why would you do that? Why don't you just go and buy some steak from down at the local store? So there were quite a few things that they were looking at. We in general, within that division, were trying to sort of find ways to do things, use biotechnology in a way that had not been used before. And so that was that really got me to start to think a little bit outside of the box and think about how you could then turn something from an idea that people get, oh, that'll never work, into, well, actually, I think I might be able to get that to work if we do this or we do that. And that's something that has stayed with me through my entire career. I am never put off by the fact that somebody says, oh, that'll never work. Oh, that's going to be too hard.
Absolutely.
I think, well, no. Let's think about that. Let's think if there's another way to do it, a better way to do it. And that's kind of has influenced my career choices over the years as I've gone through that.
I'm sure, and I guess there's an element of, as you said, timing and serendipity with everyone's career, right? But to join a company with the sort of resources that ICI had, but in a phase where they were really looking to experiment and figure out what to do and ask new questions and find the answers it's quite a unique period, isn't it?
Yeah. And so that was very that's quite very educational, actually, for me. Which was great. And then while I was there at I guess I was with ICI longer than ICI existed, actually, because it went on to become Zeneca and then AstraZeneca, and then they spun out a company, Avicia. So all the time while I was there, and during my time there, while I was, I guess when it was in Zeneca or AstraZeneca, another interesting opportunity came along. Where we had set up a collaboration with the Defence Science and Technology Laboratories, DSTL, which is based down at Porton Down, and they were looking to develop some new next generation vaccines. So they had some old technology vaccines for things like anthrax vaccines and the like, that basically took ground up anthrax and neutralized it and then used that as a vaccine. And they were trying to find a much sort of cleaner, more robust, more reliable way of producing vaccines using recombinant technology. And I got involved with helping them with that to develop new vaccines. And that was sort of in the late 90s. and about sort of 3 years in, we had some success in terms of developing these second generation vaccines, and we're getting ready to take these into a phase 1 clinical trial, and then again, it's just things that happen in the course of your career that just you can't ever plan for, but actually influence quite significantly what happens to you. So 9/11 happened, and at the same time 9/11 happened, there were the biothrax, the anthrax attacks in the US, and so there was a real uptick in the interest of producing new generation vaccines that could be easily produced reliably for larger populations, so you could protect large populations if you needed to, and that really changed the whole funding landscape as well, from a few millions here and there up to hundreds of millions to billions are being poured into how do we secure ourselves from biodefense issues. So I happened to sort of ride on the crest of the wave, I suppose, since we'd already developed 2 vaccines, one for, at the time, for anthrax and another one for plague. All the nice things in life.
Yes, absolutely.
And they both were made to GMP, were about to go into phase 1 clinical trials, and then there was this real sort of surge of funding that came through that enabled us to move a lot faster, get a lot more done. And that, as a result of that, I actually, myself and another, so I was a scientist, and there was a more sort of commercial business development person, got together and set up a little subsidiary in Zeneca or AstraZeneca, whatever we were at the time. So the 2 of us basically grew that business, that subsidiary, to about an 80 or so strong set of employees, and we had 2 products that were in late phase 2, going into phase 3, and at that point, it was Avicia at that time, when we matured the business, they decided to basically put the business up for sale, and see if somebody else wanted to take that forward through to the next stage. I think really, the main interest in biodefense was based out of the US, and so the obvious place to sell the business was overseas to a US biotech company, which is what they did. And I was the chief scientific officer at the time, so I then had another fork in the road in my career. I could either decide to go with the sale and go out to the US, or follow a different path and leave biodefense and do something different. So I opted to go with it and go out to the US, and then spent the next 11 years working in the US in various with various companies there, Emergent being one of them, United Therapeutics, and the last of those companies there before I came back to the UK was the Sabin Vaccine Institute that we talked about earlier. And it was actually so that was all going well, and I was really enjoying my time out in the US. And then another opportunity came up where you have that fork in the road again, and you can stick with the work I was doing with the Sabin Vaccine Institute, which I really enjoyed, and I thoroughly liked what the Sabin Vaccine Institute were trying to do. My job was great. The people I worked with were really good, and I was approached about coming back to the UK to help set up a new venture there, a sort of vaccine manufacturing innovation centre. I was like, well, that's quite interesting. So I went home and I sort of talked to my wife and my daughter about it, and I'd love to say that, I analysed everything and decided this is a great career path to go down. But actually, the truth of it is, they turned around and said, "You've got to take that job because I want to go and live in the UK for a bit." So off I went back to the UK, and
yeah, exactly, yeah.
That gave me the opportunity to come back and do another slightly different
Yes,
aspect or facet to my career. So rather than doing product development, I was now providing a service out to other people to help them with their development. So the whole purpose of VMIC was that it was there to provide early stage developers, particularly academics, with the opportunity to be able to take their products into GMP and to then get into the clinic. And that's has always been quite difficult to get, and quite expensive. GMP is expensive anyway, but it's particularly difficult when there's a shortage of supply in terms of the ability to be able to easily do that. So so being able to sort of set up a center that was going to be primarily focused on helping early stage translational science of taking sort of academic and early stage biotech startups and sort of foster them and take them through that process and be able to get them into the clinic was very exciting. So I came back to the UK, decided to join. It was based in Oxford, or just south of Oxford, and
this was 2019. You took that job on?
This was summer of 2019. So I had 6 months of 6 months of relative sanity, and then of course COVID came along, and the whole world went nuts. And right at the center of that, there was the UK, like many countries, was very concerned about how they were going to develop and manufacture a vaccine. And as we later saw, actually, what happens when something as significant as COVID comes along, that borders start to close down. People were not allowing vaccine or components to ship between different countries because they wanted to keep it to themselves. So the UK, alongside many other countries, wanted to have its own facility, wanted to have that capability. And so they turned around to a very small vaccine manufacturing innovation center and basically said, make it bigger and do it faster. We went from a relatively small program, small facility that was going to be able to make upwards of 2 to 3 million doses of vaccine a year, to being able to have a facility that can manufacture 70 million doses a year. So that's quite a significant increase in size. And of course, you're trying to do all of this increase in size at the very time where everything is sort of shut down. You can't get supplies. You can't get builders to come in and do things. You can't recruit people easily. And we're all learning about Teams for the first time and how to meet people remotely. And so that was I guess a combination of an extremely exhilarating and extremely stressful time.
Yes, I'm sure.
And I still can't make my mind up which it was. It was both.
No, absolutely, I'm sure. And I think it was an interesting time in that it showed what was possible, right? In terms of the response. And it must have been fascinating to see that come together.
It was. I mean, if you think what we achieved in that time, we essentially took a concept, and I was based down here in Oxford, helping to build the facility, and so we had close associations with Oxford University and the development of their ChAdOx vaccine that went on to become the AstraZeneca vaccine. So we were developing all of that as we went along, and typically taking a vaccine from concept and early stage data through to a licensed product that you're putting into millions of people, about sort of 10 years, 10 or 15,
Yes.
10 years on average, it was done in 10 months. I mean, that's just unheard of. So, and why did we manage to achieve that? It was really sort of because everyone knew that nobody wanted to be stuck in lockdown for forever.
No,
so
of course.
we had to get something done. And so everyone really focused on coming together and working together. So it was a combination of, industry and the government and academia, the sort of holy trinity, all brought together and focused on how do we do this in the shortest possible time and get a product that works and it's safe and that we can give to people and protect them and stop this terrible crisis and epidemic that's going on.
Yes.
So it was. I'm going to use the same words again. It was sort of it was really exhilarating doing that, but it was incredibly stressful as well.
No, I'm sure. And to a great extent, obviously, life has returned somewhat to normal now, right? And there will always be things that have changed. And I think, particularly in people's attitudes towards certain things. But have you seen any lasting effect in terms of the positives that came out of it, in terms of the ability to collaborate and the ability to move quickly and the ability to think of new ways to approach this whole challenge of regulatory approval and
yeah, no, I think absolutely. I think the fact that it was achieved and we successfully produced a vaccine within 10 months that went out into millions of people showed that it could be done.
Yes.
And lots of things that dogma said could not be done, you're going to take 2 years before you can go, complete your first phase 1 and another 3 years to do your phase 2. And then another 3 years for that all just went out of the window. It showed that you could actually do this a lot faster. And needs be when the devil drives and all that, as they say. So we had to work as safely and carefully as we could, but we had to also do things that wouldn't normally have you wouldn't normally have been able to do. And I think what that showed was that actually you didn't have to sort of stick rigidly to the framework that had been used before. And I think the regulatory authorities also, they were part of that process in helping to bring a very big part of the process in terms of bringing vaccines to market. So they too learned from that process. And yes, there were certain things that they still want to see done and have done that takes more time, but they also learned sort of how we can do that more efficiently, I think. And so I think there's definitely learning that came out of it, just from a scientific and regulatory perspective, to show, there's nothing like being shown that something can be done to then realize that you can do it.
Absolutely. Yes, true.
So that was very very positive. And then there were a lot of other impacts in terms of the way that we work as well. I mean, the whole sort of remote meeting thing is a classic example. People working from home, doing hybrid roles where you don't have to be in the office for 5 days a week necessarily. Some jobs you do, but a lot of jobs you don't, and you can actually do some of that work from home quite successfully, and all the systems to be able to do that were put in place. And now we all quite do that quite naturally and don't think twice about jumping on a Teams meeting or a Zoom meeting to go and sort of meet with people who are other side of the country or on another continent or wherever it might be. And it's sort of second nature now. So that irrevocably changed sort of how we work, I think, and in a positive way. And as with all these things, there is a balance to be had. So you don't want to go completely to the extreme of not meeting people. You get a lot more done, I think, and having sort of in-person, face-to-face interactions. There are certain things that just can never be substituted by doing that over Zoom or whatever. Sometimes you have to get together with people. I found particularly if you're trying to work on a particularly sort of knotty problem where you're trying to look at all different sort of aspects, being in a room with people where you can sort of bounce ideas around is much easier than it is trying to do it with 16 people on different boxes on your screen. So there are times and places where it still is better to be able to meet in person and do things in person, but there's also a lot you can get done remotely. So
yeah, no, absolutely. I think that certainly we were heading that way anyway, right? But there was a lot of resistance that got wiped out, and things certainly got accelerated as a result. So Matthew, we've covered a lot of ground talking about your career, because you've also covered a lot of ground. But if there were, and we may have talked about some of this already, but if there were maybe one or two things that you would pick out as, I suppose, things you wish you knew when you started out, or if you were talking to someone who was early in their career now, the advice that you'd give them, what would those one or two things be that you think have been really important lessons for you in your career?
I think, well, for me, it's all. I think it's important to always be curious, and be open to opportunity. And I guess sort of earlier on in my career, you're looking to other people to sort of how other people have done things, and this depends on your character, I suppose, but quite often people have a sort of reticence to try something new or do something else, and usually the underlying reason for that is they're worried about, well, what about if it doesn't work, or what about if it fails? And that should never be the reason to not do something. So you should always, I mean, you should always look at the risks and look at pros and cons of doing something, but don't let the fear of failure drive your decision to not do something. That should never be the factor. It should be other things. And I think that's a really important lesson that you learn as you go through that career. And then we've talked about a number of other things as well. I think the other thing is, don't underestimate the importance of your interactions with people and the environment that you work in, as to how much that can really drive success. When you get a good team of people together who are firing on all cylinders and working synergistically, you can get so much done. It's just amazing. And I guess the last thing there is, you should not lose sight of the fact that it has to be fun. You've got to enjoy what you're doing. Absolutely. And it's, I'm a realist. It can't be fun all the time. There's some miserable things that you have to do in your career, in your job, and from day to day and week to week, that you dread doing or you don't want to do. And everyone has to do some of that. But overall, in balance, you have to look at something and be happy about where it's going, and happy about how you're doing it, and happy about who you're working. If you've got that, then you're golden.
No, absolutely. Well, Matthew, thank you so much for joining us. Thanks very much for listening. Careers in Discovery is sponsored by Singular, helping you to build a brilliant biotech company. Biotech leaders spend far too much time, money, and energy on hiring and people issues. Head to www.singular-biotech.com to learn how you can recruit and engage your team more effectively, so you can focus on developing medicines, treating patients, and saving lives.